I looked at the list of people who have signed up for this one, and it's mostly, not surprisingly, Northeasterners. You'll know more about why that's not surprising after you hear from our speakers on the spongy moth in our yards and forests. Our speakers tonight are both Cary emeritus scientists. Now, emeritus means retired, but don't hold your breath. I don't think Cary scientists retire very well. Clive Jones, who will be our first speaker tonight. Clive, you were original NYBG, right? You got here before everybody else. And Clive has been thinking about spongy moths on and off. I had to make sure I cleared this for over 30 years. And Charlie Canham, who is also a emeritus scientist. We're going to do a hybrid lecture, so Clive will talk in real life, and then we will hand off to Charlie, who will talk, and then we can answer questions. Charlie has also been part of a project that I think Clive will discuss in some detail, but as with everything at Cary Institute, we're really interested in how things work, because we like to say you can't understand it you can't solve problems unless you understand them. And so Clive and Charlie and a number of other people spent decades trying to figure out the factors that drive spongy moths, although when they started, it was driving gypsy moths. They outlived the name and the project. I will just pass to Clive now with the very quick comment that about 2 years ago, I called Clive because the spongy moths were getting worse and worse, and I thought I'd better know what I'm talking about, so I knew who to call, and I called Clive, and I left New York City, and 49 minutes later, I arrived where I was going, and our conversation was just ending, and I thought, what a great thing we can do at Cary, a Cary Science Conversation, a Friday night at the Cary. We could do something about this because so many people seem to care. So, Clive, it is a pleasure. I'm glad we are doing that, and Charlie, welcome, and we look forward to hearing from you in a little while.
Well, thank you, Josh. I won't talk for an hour, but I'm going to talk about the spongy moth in our yards and forests. And what I'm going to do is tell you where the spongy moth comes from, when it was introduced, and what happened next. Then give you a little bit of background in relevant life history and ecology that will help you understand what I then say about the causes of outbreaks and their collapse. Then I'll talk a bit about why this current outbreak and what you can expect next and in the future. And then a little bit about what you can do about the moth. Then I'm going to pass it over to my colleague Charlie Canham, who will talk about what will happen to the trees in the forest. So the spongy moth is Lymantria dispar, and it's native to Europe and Asia. But in North America, it's now classified as a subspecies Lymantria dispar. It was introduced in Medford, Massachusetts in 1868 or 69 by Etienne Trouvelot, a French amateur entomologist and erstwhile entrepreneur, who thought he could help develop a silk industry in the cold Northeast by crossbreeding the winter-hardy spongy moth with a cold-intolerant Asian silk moth. Now, that wouldn't have been possible, but nevertheless, he got some egg masses, opened the box, put them on the windowsill of an open window. When they blew into his garden, he couldn't find them, but he did report it to the authorities. This is the Medford area 20 years later. The crosshatch areas in the middle are where the defoliation was complete, and the 200 square mile boundary is where the spongy moth was recorded as being at least present. So how did it get from Trouvelot's backyard to that 200 square mile area when the females of the North American subspecies are flightless? Well, there are two ways. First, the early stages of the caterpillars balloon on silken threads. Most of them go a few yards from tree to tree, but occasionally they get caught up in the wind and go much longer distances. And the second, and most important, is people moving around on cut timber, on garden furniture when they move homes, and on the underside of vehicles such as their RV that they take on holidays somewhere else in spring. So it's come a long way in 150 years, And this animation from the Forest Service will show its spread, more or less annually, but there are breaks in the data. As you can see, it's spreading steadily west and south and north, but for some reason not into Canada. I don't know why. No. That's because it's US Forest Service data. All of that spread is due to ballooning and people moving egg masses around. And it's still spreading. Okay, so this spread has been accompanied by periodic outbreaks, defoliation, and collapse. And by that, I mean there's rapid increases in moth density, and then they come back down again, with defoliation at the peak. Now, these outbreaks are not unique to North America. They occur in the native range. This is Serbia from 1945 to about 2015. It's in an obscure axis there of infested area in hectares times 1,000, but that peak there represents about 2 million acres defoliated. Outbreaks occur about, and it's really approximate, 10 years on average, and that's really an average. They can be relatively synchronous over very large areas and can lead to extensive defoliation, such as over 9 million acres in the outbreak of 1981. This is going to show you another Forest Service animation. The moving red blobs you see here are defoliation. The data go from 72 to 2007. Keep your eyes on the 80s as we... And then, again, in the 90s. And then, as this outbreak wanes away, things stayed pretty quiet up until relatively recently. Okay, so now a little bit of relevant life history and ecology that'll help you understand what I later say. In late summer, the females emit a chemical signal, a pheromone, that attracts the males that develop a little earlier over quite long distances. They mate. She lays an egg mass containing a few hundred eggs protected by scale hairs. In low densities, these egg masses are laid at the base of trees, but as density goes up, egg masses get laid all the way up the tree and on the side branches and on your garden furniture and on the underside of your RV. They overwinter as egg masses, and they hatch out in spring more or less synchronously with bud break of their preferred host, oaks. And they go through 5 stages or instars for males, and 6, including the big one at the bottom, for females. The early stages, the first, second, and third instars, balloon on silken threads, as I mentioned earlier. As the density of spongy moth goes up, a greater and greater and greater proportion of the larvae start to balloon and disperse. In low densities, the later stage larvae spend the day at the bottom of the tree resting, going up to feed in the canopy at night. But as density rises, more and more of the caterpillars spend all their time in the canopy feeding non-stop, and when the food has run out, they move across the defoliated canopies and all over the ground. They then pupate, which takes a couple of weeks at the end of summer, the smaller males at the top and the larger female beneath. And the cycle repeats itself after the female emerges, mates, and lays eggs. So next, I want to tell you a bit about natural enemies of the spongy moth that don't really make a big difference to outbreaks or their collapse. They do kill the moth. They're native and species we introduced to try and control the moth. They are predators, parasites, pathogens, and collectively they attack all the life stages. But their failure to kill doesn't cause outbreaks, and at best, they can help an outbreak that's already collapsing collapse further. Why is that? Well, they kill too few, there's better food elsewhere. They're not very abundant, so even if they do kill moths, they don't kill many. They're reasonably abundant, they do kill moths, but they don't increase in abundance as the spongy moth is rising in density. Or they do all of the above, and they do increase, but they do so too slowly to be able to catch up with and overtake and bring down a rapidly rising moth population. So they include birds such as this cuckoo, one of the few species in North America of birds that will eat hairy caterpillars. Most will not, but cuckoos are not that abundant. They include most small mammals, with one major exception we'll talk about in a minute. So the smoky shrew, for example, will eat larvae and pupae that it finds on the ground, but they don't spend most of their time on the ground, and shrews are not that abundant. Chipmunks can go up trees and are very abundant. However, they don't eat larvae or pupae. And finally, there are things like caterpillar hunter beetles and ants and spiders that will eat both larvae and pupae, sometimes in reasonable numbers, but in general, most of those species do not increase in abundance as the spongy moth increases. Then there are parasitic wasps that lay eggs inside an egg mass, and that parasitize caterpillars, sometimes again in reasonable numbers, and they do increase in abundance over time, but they lag well behind the spongy moth. So at best, they can come in towards the end of an outbreak. And finally, most pathogens, with two exceptions we'll come to in a minute, have no effect. So this is the naturally occurring Bacillus thuringiensis. It occurs in soil, so it has little or no contact with the caterpillars. And more importantly, in order to kill a caterpillar, it has to be eaten. And caterpillars don't eat soil. This is in contrast to the BT that you spray on the leaves of trees in high doses, which does get eaten and does kill caterpillars. So let's turn then to what does cause spongy moth outbreaks and then their collapse. There are two causes. The first cause is high female fecundity, which imbues the moth with the capacity for rapid rises in density. So imagine that every egg in an egg mass survives to adulthood, and there are about 500 eggs per mass, and about half of those are females. Both of those are reasonable numbers. Then the maximum theoretical rate of increase of the spongy moth is 1 times 500 times 0.5 equals 250-fold each year. The maximum we've actually observed in the field is about half that, 125-fold. So think about that. In year 1, you have 1 egg mass. In year 2, you have 125. In year 3, you have over 15,000 egg masses, and that is more than sufficient to cause complete defoliation, which is increasingly likely once you get above 2,000 egg masses per acre. The highest we've ever recorded before collapse of an outbreak was 6,000 masses per acre. So preventing outbreaks requires keeping moth density really low. Otherwise, it's just going to escape and get away. And the second and important cause of outbreaks is mouse population collapse. This is the white-footed mouse, Peromyscus leucopus. It's the most abundant small mammal in our forests, and it is a voracious predator on moth pupae. Not larvae, not egg masses, just pupae. And as you can see, they can go up trees to get to pupae. This shows you how long a female pupa is going to survive in the forest as a function of mouse density, the percent of pupae remaining over time. It takes 12 to 16 days, about 2 weeks, for a female to complete development, emerge, mate, lay eggs. And this particular study is done with freeze-dried pupae, affixed to burlap panels using beeswax with the panels at the base of the trees. The beeswax leaves characteristic marks of who attacked the pupae, in this case, the tooth marks of the white-footed mouse. We've also done this experiment with living pupae. We get the same result. We remove the females and the egg masses. And we've also done experiments in which we live trapped mice on large areas on large grids when moth density was low, moving them to an area of Cary forest quite some way away, whereupon the pupal survivorship went up, the egg mass density went up compared to the control grids where we didn't remove mice. So what this shows is that in 93, where you have 7 mice per acre, a moderate mouse density, none of the pupae survive or persist, because they're freeze-dried in this case, beyond 8 days, which is well short of the time it takes for a female to complete development. In contrast, in 94, when you have 1 mouse per acre, 50% of the pupae are still remaining 18 days after deployment, well beyond the time it takes for a pupa to develop as a female and lay eggs. So what this means is that moderate to high mouse densities keep moth populations low, can drive them to even lower levels, and within reasonable limits can prevent a moth population that started to rise from rising further. But low to very low mouse densities allow outbreaks to start. Now, mice eat moth pupae, but moth pupae do not directly affect how many mice there are. And that's because mice are omnivores. They eat insects and worms and the eggs and nestlings of ground nesting birds and a tremendous array of different species of seeds. So moth pupae are just a minor part of the diet, a 2-week snack for mice that has major consequences for moths. Instead, the number of mice is determined by the number of acorns produced the previous fall, what's called masting. So this shows you 5 of over 30 years that we've been doing this at Cary, on 2 of 6 grids that we've been doing this on. And mast means some to many acorns, and no mast means none. Okay, so in moderate to large acorn crops, as you see here, many mice survive the winter. They start reproducing in winter, late spring, packing in an extra mouse generation so that by the time the moth pupates, there's high densities of mice. In contrast, when there's no mast or very low acorns, few mice survive the winter. They start reproducing much later, and you get very low mouse densities by the time of moth pupation. We've seen this basic pattern for 30 years. We've also done some experiments at low moth density where we, in a non-acorn year, we dumped about 1 million acorns into some of our grids, whereupon the next summer the mouse density was increased, whereas in the controls where we didn't add acorns, it was not. And at the same time, the increase in mice brought down the pupal survivorship and the egg mass density. And this basic pattern between acorns and mice has now been demonstrated in a number of locations by other people. So the moth doesn't directly affect the number of mice, but oak defoliation can reduce acorn production. As you can see here, with moderate to high and then low defoliation in 80 through 83 and no acorn production during that period. So what that means is that by suppressing acorn production due to defoliation, they can suppress mouse populations and thereby increase the risk of another moth outbreak. Okay, so let's now move to the causes of collapse. There are 3. And the first is the fungus Entomophaga mimega, indicated by this head-down desiccated cadaver to the right of the pupa there. It was introduced from Asia in 1910, but failed to establish, but then it was reintroduced in the late 1980s and is now quite widely distributed in the Northeast. And if it's established in an area, it will persist for reasons I won't go into. It kills some larvae each year at low moth density. And it can kill many larvae within a year at moderate to high densities, and as a consequence can curtail outbreaks with low to moderate defoliation before collapse of the moth population. We don't understand exactly what causes this mass mortality, but we do know that it's more likely to occur when larval density is moderate or greater and spring is both wet and cool, where cool means there isn't a period of days, of 4 or 5 days, where the temperature is more than 80 degrees Fahrenheit, which inhibits the fungus. Second is the nuclear polyhedrosis virus, or NPV, as indicated by this soggy inverted V of a caterpillar that just died from the virus. It's naturally occurring, it's always present, it's mostly sublethal in low moth density populations, but it's highly lethal at high moth densities. And it's the primary cause of collapse of Gypsy, spongy moth, beg your pardon, in North America. So why this transition from sublethal to lethal? Well, it turns out that increasing moth density increases larval competition for food and for resting space, which increases the stress on the larvae, which decreases their immunity, which increases their susceptibility to the virus and viral mortality. And the third and final cause of collapse is food limitation. You run out of food and you can't complete development and produce eggs. It's also true, of course, that later stage larvae do eat non-oaks, but their survival and their fecundity is much lower than on oaks. So food limitation can and does bring about collapse, and as I pointed out, via the stress it causes on caterpillars, it can boost the efficacy of the virus. I want to end this session on collapse and outbreaks by showing you all the players together over 30 years at Cary for Julie Hart's benefit. This shows 1980 to 2010 egg masses per hectare on the left and per acre on the right. Each of these divisions is on the logarithmic scale is 10 times more than the division below it. The lowest density we've ever seen was 1 egg mass in about 25 acres of forest. And the highest density we've ever seen was 6,000 acres just before the collapse, 6,000 per acre just before the collapse. Each of those data points is the year in which the egg masses were laid, so all the action I'm going to talk about occurs in the spring, summer following that year of oviposition. So the outbreak ongoing in 1980 led to extensive defoliation. That outbreak collapsed due primarily to food limitation and the virus, with some invertebrates playing a role towards the end when the outbreak has already formed. There was no fungus. Hadn't arrived. An outbreak began in 88, probably due to acorns and mice, based on what we know now, but we didn't start monitoring mice and acorns until 91. That was curtailed with low patchy defoliation of about 20% due to the fungus. The virus was detected but didn't play a role. From then on, this entire low-density oscillation from around 1 egg mass to 10 egg masses per acre was due to mice and acorns. When acorns failed, mice failed, moth went up. When acorns were produced, mice went up, moth came down. Throughout that period, the fungus was always detected, and occasionally you could see that the virus had killed 1 or 2 caterpillars. All right, so let me talk about the current outbreak. So this shows a acorns, mice, fungus, virus, and defoliation in egg masses from 2019 to 2023. This is reconstructed with the help of colleagues here at Cary. We have data on acorns and mice, but the fungus, virus, defoliation, and egg masses are just based on observations. So there was a very high acorns in the fall of 2019 led to a very high summer mouse population, likely the conditions that continue to maintain the low density of spongy moth that has been here for a long time. There was low acorn production in 2020, which led to very low mice the next summer. These are the conditions in which outbreaks get started, and based on the trajectory subsequently, this is when we think it started. In 2021, there was a moderate acorn crop and a reasonably high mouse population, but it looks as if it was too late. Our mathematical models show that if the moth density starts to get above 10 masses per acre, mice can no longer control it. So that's what we think happened here. We detected the virus and the fungus, but they didn't really do much, and there was an observed increase in egg mass density. In 2022, very low acorns leading to very low mice the next year, but by then, it didn't help, but by then the moth has already escaped from mouse control. Again, the fungus and the virus were detected, but had no real big influence. Spring that year was drier than usual, so the conditions may not have been as conducive to the fungus as one would hope for. And as a consequence, there was 50% defoliation and another increase in density. This is the defoliation on the tea house hill of about 50% in June, and complete refoliation by the end of July. The conifers at the top of the tea house hill you see there were unaffected. So what next? So the best guess for Cary is that 2024 we will have an NPV-induced collapse, well, plus or minus the fungus, depending on the weather. Whether or not there'll be a lot or a little defoliation will really depend on how fast the virus moves through the population. If it moves slowly, there'll be complete defoliation. If it goes through fast, there'll be incomplete defoliation. And if that doesn't happen, and the density continues to rise or stays at the level it is, then a collapse in 2025 has a much higher probability, because by then food limitation and stress will be kicking in. After that, continued to collapse to low density, a period of unknown duration when the moth will be rare due to the mice, but you should expect outbreaks in the future, with the severity being determined by the efficacy of the fungus, and which will in turn depend more or less on whether we get more or less wet springs. I want to sort of end this section with what can you do about the moth. So this is a long list of interventions, with those you can't do in New York crossed out in red. You can read all about them. What it really tells you is when you might expect them to work. So I'm going to give you 4 guidelines. Guideline 1, outbreak stage as years after mouse failure. So early on, there's low to moderate density. There's potential for having some influence. The egg masses are accessible because at the base of tree, you could remove them or treat them with dormant oils. Larval immigration via dispersal occurs, but is quite limited. So sticky tape and burlap bands around trees also will work. In the middle of an outbreak, when you have moderate to very high density, you have very limited options. You can't remove the egg masses because now they're all over the trees, and you can't treat them with dormant oil. The larval immigration via dispersal is high to very, very high, and a lot of the larvae stay in the canopy anyway, which means that sticky tape and burlap bands play little or no role. So your options are BTK, but you can't use it near water. It requires carefully timing 2 applications, and you have to hope it doesn't rain just after spraying because that washes the BT off the leaves. Or for that matter, it's not too sunny because the UV inactivates the BT too. It's the lower risk of all of the options. The effects on non-target Lepidoptera, which is the only other sort of tree-feeding insect, are limited because most of those hatch and emerge and develop much later in the season. Acephate systemic tree injection is still allowed. It carries a risk of girdling the trees and killing them. You can only use it on non-bearing trees, which means they mustn't fruit, flower, or produce seed within a year of treatment. It kills most, if not all, leaf-eating insects, and there is a risk of non-target effects on invertebrate and vertebrate predators that then eat poisoned insects. Late in the outbreak, the density is falling, or at least not rising. You've probably got a lot of fungus and MPV. There are epizootics going on. The outbreak's ending. You don't need to intervene. Second, what's your goal? A fully accessible tree or a sapling is much easier to deal with than a lot of tall trees, which is much easier to deal with than a big chunk of forest. The third, the area of intervention. So small areas are much more viable to have an influence on than very large areas, but you have to bear in mind that surrounding areas can overwhelm whatever local influence you're having due to immigration and dispersal. Fourth, and as a consequence of that, location really matters with respect to the surrounding forest. So isolated trees you might have an influence on. It's much more viable than trying to deal with the ones next to the forest, and certainly way more viable than those that are deep in the forest. I want to just point out that Forest Service and New York State DC no longer intervene except into some very special circumstances. And as a consequence, the spongy moth joins a really long list of introduced forest pests and pathogens that we are unable to control once they arrive. And so with that, I'm going to pass over to my colleague Charlie Canham remotely, and who's going to talk about what will happen to the trees and the forest. So over to you, Charlie, when I press escape. Magic.
Am I live, Leslie? Okay. I'm going to share my screen and just go to my slides. Okay, spongy moth, as Clive has said, is just one of a much longer list of the introduced pests and pathogens that are arguably the most pervasive and significant threat to eastern forests. This is a slide I typically use when giving talks about the future of eastern forests, and you'll notice that spongy moth is not actually on the list of the pests and pathogens that I consider the greatest threats. In fact, in the chapter in my book, Forests Adrifts, about the future of northeastern forests, I call out spongy moths as being distinctive in that defoliation by the caterpillars is rarely a direct and immediate cause of tree mortality. I'll explain why and mention an exception to that generalization in a minute. Compare that to the first 6 pests on this list, each of which has caused decimation of the host tree species they attack. I'm sure many of you have dead or dying ash trees on your property or in your neighborhoods. Emerald ash borer has caused the most rapid decline in native tree species of anything on this list, in part because the insect is such an effective disperser. Basically, all of the more than a dozen ash species in North America are currently listed as critically endangered by IUCN. I put spotted lanternfly and beech leaf disease on this list, even though it is far too early to know just how destructive these will be. And that highlights one of the most enduring challenges of dealing with the emergence of new pests and pathogens. It can take years to do the research, first to know how serious the threat is, and then to find the ways to combat it. The work that Clive has described, and not just his own work, but the work of many others, literally took decades so that we could understand the spongy moth system. And that highlights the other enduring challenge. How do we stop the emergence of these threats, particularly new pests and pathogens? And I'll talk more about this in a minute. But the bottom line is that we now know that aside from the very colorful history of spongy moth introduced through entrepreneurial misadventure, there are really two main routes by which they are introduced. For wood-boring insects, the vectors through solid wood packing brought into the country when goods are imported. Other insects and diseases hitchhike on the importation of live plants. So it's no surprise, but probably shocking to you, the degree to which trade through Northeastern ports has led to such a concentration of non-native pests. And unfortunately, once here, they inexorably spread across the country. Let's see. So, spongy moth has been around for long enough that there is a reasonable body of evidence about its impacts. And Clive has described the incredible detail we have from his work on the ecology of the insect and the factors that control outbreaks. We frankly have much less detailed information on the impacts of defoliation on our tree species, but the bottom line is really very clear. Complete defoliation in early summer, even for 2 years in a row, kills very few trees directly. As horrifying as it is to witness the defoliation, to understand why trees are so resilient to defoliation, you need to understand their carbohydrate economy. Basically, photosynthesis during the growing season produces the sugars needed to produce new tissues and the energy those tissues need for their metabolism. But the even more important outcome of a good growing season is essentially the profit left over after meeting those immediate needs. And that profit is in the form of sugars and starches that act as reserves for use next year. As an aside, those carbohydrate reserves are also critical for the cold hardiness of the trees. They essentially act as antifreeze. And next spring, those reserves are mobilized to produce the new wood needed to conduct water to new shoots and leaves, as well as provided the energy needed for the first stages of stem and leaf growth. And then as the season progresses, the leaves are recouping that investment in reserves, and the time required for that varies enormously. But there's little question that complete defoliation in June by spongy moth happens right around the time when the leaves would be starting to produce a profit and replenish reserves. Luckily, it's really very clear that the spring flush of growth and mobilization of those reserves doesn't completely use up last year's reserves, and the trees very rapidly produce new leaves, as I'm sure most of you observed on your trees. And understanding the role of these reserves helps explain, one of the exceptions to this generalization that defoliation rarely kills trees. The Canoe Hills here at the Cary Institute had a really abundant understory of hemlocks 40 years ago. Hemlocks are one of our most shade-tolerant trees, and over time they would have actually grown up and replaced the overstory oaks. But when we sampled the Cary forests in 1984, right after I joined the scientific staff, virtually all of those understory hemlocks were dead, and we assumed that this was in fact a direct result of the defoliation in 1980, 81, and potentially 82. Now, hemlocks are evergreen, and those needles actually last 2 to 3 years, but they're also an expensive investment of carbohydrates produced. Those tissues are very dense, and by living longer, they can still generate a net profit, but in the heavily shaded understory, that profit is actually quite small. And we assume that 2 or more years of defoliation basically wiped out their reserves and killed them outright, either because of hydraulic failure due to inability to produce new wood or an inability to fight off root rots and other pathogens. The hardest thing to quantify is whether there are long-term residual impacts of the stress of several years of spongy moth defoliation. In general, stresses on trees compound, and ultimately the combination can make them vulnerable to some other more direct cause of mortality, say something like a bark beetle attack or a root rot fungus. We recensused the Cary forest plots in 2006 and found an unexpectedly high mortality of chestnut oaks, not catastrophic, but just higher than we would have expected. Chestnut oaks are one of the dominant oaks here in the mid-Hudson Valley on hill slopes. In the 25 years after that 80, 81, 82 defoliation, saw a number of modest droughts. There were some years with high ozone levels before the Clean Air Act amendments of 1990 fully kicked in. But it is reasonable to assume that spongy moth defoliation contributed to the higher rates than expected of chestnut oak mortality. So when I think about the impacts So when I think about the impacts of a particular year of defoliation, what I really want to know is how favorable are growing conditions for the new flush of leaves during the rest of the summer. Now, 2 summers ago, 2022 was the worst summer drought in the Hudson Valley in over 60 years. And I would expect that trees defoliated earlier that year, in the spring, June of 2022, struggled to replenish reserves over the rest of the summer because, frankly, the leaves had to shut down their stomata because of the low soil moisture and therefore couldn't photosynthesize. And that's why I was frankly so happy that this past summer was one of the wettest summers in decades. The trees in my yard had their first complete defoliation this summer, but I feel pretty confident that the heavy summer rains put them in decent shape for what will inevitably, given everything Clive has talked about and the thousands of egg masses on my trees, is going to be a second year of heavy defoliation this spring. So this very long-winded detour into plant physiology leads to the one general recommendation I have for helping your trees weather a spongy moth outbreak. It's really very simple. If it's dry in midsummer after defoliation, think about watering the rooting zone under your trees. I don't generally favor fertilizing because it's easy to over-fertilize and potentially make the trees more tempting targets to other insects. But soil moisture availability has a really strong positive impact on photosynthesis and the ability of the trees to replenish their reserves. So Clive ended his comments by pointing out that spongy moth is one of this long and really quite depressing list of non-native pests and pathogens that we have been unable to control once they get established. And as I've said before, I consider these pests and pathogens to be the single greatest threat to eastern forests. So I want to end my comments by talking about a recent initiative called Tree Smart Trade, started by our late dear friend and colleague Gary Lovett. Many of us here on the staff, Clive, Kathy Weathers, myself, and particularly Gary, have spent years studying the ecological and economic impacts of forest pests and pathogens. Around 10 years ago, Gary convened a workshop with experts from the US and Canada on this issue and led the writing of a very important paper that outlined not just impacts, but policy options. And to his everlasting credit, Gary realized that just writing another paper for the scientific literature wasn't enough. And he and others devoted themselves to this new program they called Tree Smart Trade. This was a bold thing for a research scientist to do, and it meant Gary started spending a lot of time meeting with industry groups and traveling to Capitol Hill. The bottom line is that the federal agency charged with inspecting and stopping the importation of new pests and pathogens, APHIS, the Animal Plant Health Inspection Service, it's an agency in USDA of really dedicated, very competent people, but woefully underfunded, given the flood of shipping containers and live plants that enter the country every day. So we need much smarter ways of dealing with this threat. And I just want to give one example, something I consider a truly brilliant idea that Gary described in his last Lunch Bunch presentation to the staff here at the Cary. Solid wood packing material by international treaty is supposed to be heat treated to kill any insect larvae embedded in it. Treated wood gets a stamp to identify it, but there is clearly some wood that is fraudulently stamped without being treated or simply was not treated sufficiently. And there really aren't any government programs that effectively police the stamping process. Now, if a container ship arrives in a U.S. port and an APHIS inspector finds evidence that an insect has emerged from the wood en route, the fully loaded ship is denied entry and sent back to sea. This is an enormous cost to the shipping industry, even given the very tiny fraction of shipping containers that can be inspected. So Gary met with shipping industry leaders, and together they realized that they needed some sort of commercial private enterprise that would, for a fee from the shippers, guarantee that the solid wood packing was truly all treated. These new entities would take on the liability if fraudulent packing material got onto the ship. So it would be in their interest to provide effective policing of the treatment program. And it's this kind of creative thinking that the Tree Smart Trade program can provide. And this is the kind of legacy that any scientist can be proud of. And with that, I'm going to stop my remarks and try to leave some time for questions. So I will stop the share and turn it over to Josh.
We can leave Charlie up there. Yeah, it's fine. So we're going to do virtual and in real life questions as a way of balancing the hybrid nature of this. If you've got a question, do we have microphones? No microphones. So ask the question. I'll repeat the question so that people, both in the virtual audience and Charlie, can hear it, and then we'll go on. Please make sure, I always say this, please make sure to ask a question, not make a statement, and only one question per person, because we have, despite the brilliant and incredibly time-conserving presentations, we only have about 12 minutes for questions. Ma'am.
What influences acorn production?
So the question is, what influences acorn production? Charlie, Clive's pointing at you, so can you, handing off, next one's yours, Clive.
Yep. Okay, so acorn production is triggered by cues from the weather. The trees flower over, depending on the oak species, a 1 to 2 year cycle. The energy, the reserves stored up over time determine the magnitude, the size of the acorn crop if it's successful. Now, acorn crops can fail for all sorts of reasons. A summer drought will cause the selective abortion of the developing acorns, for instance. But combination of basically storing up energy for several years, following the weather trigger that starts flowering is what determines the size of the acorn crop.
Great, thank you very much. Question from the virtual audience. I've been watching the infestations moving around the Hudson Valley in New York State the last few years and wonder if you have any insights on the general directions, north, south, east of west, or of outbreaks. So do they follow any patterns, Dr. Jones?
The short answer is not really. So if you recall looking at that defoliation map, you see some general patterns in which a lot of areas go off together. But if outside of that, you'll see areas winking on and off. Now, what that means is largely that the same basic dynamics we talked about earlier with acorns and mice and so on and so forth are playing out locally. There's no expectation that acorn production, even if it's synchronous over quite large areas, will be perfectly synchronous, nor that all the mice populations will be exactly the same in each of those locations, or that the habitats for them are the same. So the end result is that although this general pattern plays out everywhere, it doesn't play out in the same place and time in the same way. So it might be that it's going Connecticut to Dutchess this time, but maybe it'll be the other way around in the future.
So it is random mostly.
There is a lot of stochasticity driven, or variability, driven by the fact that, you suppress the moth population and then it rebounds. But how high did it go? How fast does it escape from the mouse? How low did the mice go that time? So an outbreak can get started here, but not here, and get started here later. So it's the same rules of the game. It's just that the rules of the game are not perfectly timed everywhere.
Thank you.
So we can't really encourage the mice, I guess, to reproduce more, because then they're going to come and bring us flies.
So I'm so glad you pointed that out. For those of you who don't know, Rick Ostfeld, who both speakers mentioned, has spent the last 35 years studying Lyme disease, which came about as a result of this project because he noticed all those ticks on ears and ears of mice and started from there. But I think the point you're making is a really good one, which is we both love and hate mice. We love them because they control the spongy moth outbreaks when they're not escaped. And we don't like them because they control the Lyme outbreaks. And it's really rare for a Cary scientist to be definitive about predicting things in the future. But Rick Ostfeld is pretty good about saying if there are a lot of acorns this year, there'll be a lot of mice next year, and the year after that, in the spring, look out. There'll be a lot of Lyme. So I think I'm going to field that question, and then I'll anything to add, Clive?
No, except, Gary, Charlie, myself, and Rick were there. We started this forest responses to stress and damage, and a lot of that work went in a lot of different directions, largely because we found out stuff.
And you hired Rick originally as a small mammal ecologist
Yes.
to deal with the mice in this
Well,
project?
I got Rick into mice early on because I said, there's some interesting work suggesting mice eat eat the moth. Are you interested?
Which is very common around here. People love to collaborate, which is surprisingly less common than you would think within institutions. So Charlie, a question for you. Are there certain forest communities that will recover better than others from this process?
Well, as Clive noted, oaks are sort of the most highly preferred food resource for spongy moth, and so it tends to be the oak forests that are hit the hardest. Oaks are also pretty resilient trees, and so we do expect much lower levels of defoliation in, say, a maple-dominated forest. And so, is that a function of their resilience or just that they're not affected? I think the bottom line is that the real answer here is if there are no other compounding, very serious stresses, all of our forests recover quite well following a spongy moth defoliation event.
So I'm going to do a quick little follow-up because there was another very closely related question. Okay, so what's killing oak forests?
Well, all sorts of things kill trees. There is no, oak mortality is not elevated in eastern oak forests over what we would expect. What's happening is that through natural succession and the lack of a historic lack of fires and suppression of fires, oaks are being replaced by maples throughout the range of oaks in the eastern U.S. As they age and die. So there's certainly, there's sudden oak death and oak wilt are two diseases that are present in various parts of the East, but they're not, neither of them is common here in the Hudson Valley. So, by and large, I consider all of the oak species in the Hudson Valley to be quite healthy. But over time, over the next 100 years, we fully expect their abundance to decline simply because they're successionally being replaced by more shade-tolerant species in the absence of, selective light ground fires.
Great. Question from the audience.
Can you tell us when's the best time to put stuff around our trees?
The question was, when is the best time to wrap your trees with burlap or sticky tape, or what time of year?
Do you mean time of year?
Yeah.
Yeah, so when the larvae hatch out in April, early May, they spend quite a bit of time before, resting and then ballooning around. So it's usually, you want to get it, the behavior for going up and down trees starts in the later instars. So probably within 2 or 3 weeks after you see hatch is probably the best time to do it. That way the barriers would be in place by the time the larvae start to go up and down trees.
But if you wait too late, it's, as it were, a fool's
Yeah, well,
errand,
that's
because they're
why I
already...
said that's, yes, assuming it's feasible and viable.
Right.
That's what I'm saying, that you should earlier rather than later. It doesn't do any harm to put a sticky band around a tree in early May. It's just that you won't be catching any larvae until later.
But to reiterate, Clive, you think this summer because...
Oh, forget it.
That's clear. A declarative statement from a Cary scientist.
I think
Forget it.
I say that guardedly because it depends on where you're sitting. Because some people, the outbreak is already collapsed in Connecticut. There are parts of Connecticut it's collapsed. You don't need to do anything. Other places, the density may not be that high. So you need to go out into yard, look at how many egg masses there are, look at where they're distributed on the trees. If you see them everywhere and a hell of a lot of them, then the chances of you having an effect are very low. If you see a few, then you have a chance at intervening.
Another question.
I live about 2 miles from here, and all of my oaks have egg masses on them. And I have a rather adolescent attitude towards what to do about it. I take a propane torch and I flame them for about 5 seconds each. It seems to be sufficient. From my point of view, who knows? But it seems to be sufficient to do away with them.
Yeah.
I don't know if I'm doing more harm than good. I'm curious what you're...
Clive, let me just repeat it. Is hitting your egg masses with a blowtorch helping or hurting your oak trees?
Yeah, well, as long as you don't scorch the bark or set it on fire. They used to use blowtorches back then. They also used to spray lead arsenate. And they used to have people climbing up to the top of 200-foot trees using ladders picking egg masses off. Okay, so if you can reach all of the egg masses, it's very easy to remove them, assuming that there isn't a vast number. So you remember what a milk gallon jug, the plastic of a milk gallon jug, you take a piece like that, you roll it into a cone, you tape it across there, you put a plastic bag on one end with a rubber band, and then the funnel part, you just scrape the egg mass, it all falls in there. At the end, you take the bag, dump it in hot spongy water, a bucket of hot water, soapy water, and it'll do a fine job. So I would, if you could reach them, I would do that. Unless, of course, you have one of those really big flamethrowers.
One more from the audience, and then I'll do one more from the virtual.
If you scrape the egg masses off, do those larvae just live in the underneath?
No, don't dump them on the ground. It's certainly true that if you break up the scale hairs, that removes some of the thermal protection. But depending on what time of the year you do it and the weather, they're very hardy. So you may, in fact, have most of them may actually hatch out. So put them into the water.
Hot, soapy water.
And yeah, leave them there for a day or so, and then you can compost them if you want.
Okay. And Charlie, you can answer the last question. What is the one thing every person watching this presentation should do to get invasive plants and insects under control? Nice, simple question.
A nice, simple question. And my answer to these sorts of questions is that there are how many billion of us on the planet, and there are so many things that need doing. Find the thing that makes sense to you and do it. There are lots of organizations that can help guide you, land trusts, conservation organizations, USDA, the Forest Service, Soil Conservation Service, etc., the Farm and Home Center. They all can provide information, and your local nature preserves can always use volunteers to help control invasives. And then support programs like Tree Smart Trade and support efforts by all organizations to stop the importation of new ones. Once they're here, we're facing millions of dollars needed to even just minimize what are already pretty devastating impacts. So find the thing that makes sense to you and do it.
I was just going to add, Tree Smart Trade, call your legislator.
That's right. So thank you very much. Before we go, I just want to say our next event is a fully virtual event on February 15th It's a Thursday. We decided not to do it on Valentine's Day in hopes that you have something better to do. We are going to hear from Cary Institute wildlife biologist Mike Fargione, who for the last decade has been putting out camera traps on our property with a scientific goal of understanding the dynamics of the deer on our property, but also it gives us real insight into all the critters that live on our property and live in the Hudson Valley. I have seen some of these pictures. I can guarantee that it will be entertaining and enjoyable, and you can even watch the show and have dinner at the same time. So that is 7 o'clock on the 15th of February, and if you sign up virtually, you'll get a link, and if you miss it, you can watch it later. Go to our website and you'll see a link to all the previous videos, and there will be one to this one. So if any of your friends ask you about what to do about spongy moths, you can say, not much, or watch the video.